This is a DNA double helix. We are going to show what is a transcription unit. The DNA consists of two strands. This is the 3' to 5' strand, and this is the 5' to 3' strand. This is a DNA double helix. Now, in the 3' to 5' strand, this is a specific sequence of DNA. This is a specific region of the DNA which is called promoter. Promoter is a gene. A gene means a sequence of DNA, a sequence of nucleotides, a sequence of nitrogen bases. So promoter represents a specific gene in the 3' to 5' DNA strand. Promoter is a specific sequence of nucleotides. Terminator. Terminator is another gene along the 3' to 5' strand, but it lies towards the 5' end. Terminator is a gene. It is a specific gene. It is a specific sequence of nitrogen bases or nucleotides. It lies towards the 5' end, and the promoter gene lies towards the 3' end. Now, in between the promoter gene and terminator gene, this is the sequence of DNA. This is the sequence of 3' to 5' strand, which is called structural gene. This is the structural gene. Promoter is a gene. Terminator is a gene. The structural gene, it is another gene. It is a sequence of DNA. It is the segment of DNA which lies between promoter gene and terminator gene. Promoter gene, terminator gene, in between the structural gene. Now, the 3' to 5' DNA strand synthesizes the RNA. The 5' to 3' strand does not form any kind of RNA. It does not act as a template. The 5' to 3' strand is not used for synthesis of RNA. It is called coding strand, but it does not code for anything. The 3' to 5' DNA strand is called the template strand. It serves as a template strand for the synthesis of, for the polymerization of messenger RNA. So RNA is synthesized by only one strand, that is the 3' to 5' strand. It is the template that serves as a template for the formation of messenger RNA. But 5' to 3' strand is not used. It is called coding strand. The 3' to 5' strand is the template because the RNA strand which is formed is 5' to 3'. So RNA must be complementary to the DNA template. They must be antiparallel. Since the messenger RNA strand is synthesized in 5' to 3' direction, the template must be 3' to 5', opposite polarity. That is why the DNA which has polarity 3' to 5' is used for the synthesis of RNA, whose polarity is 5' to 3'. So the template strand and the messenger RNA which is formed must be antiparallel. 3' to 5' template, messenger RNA is 5' to 3'. It will be synthesized from the template strand. But the 5' to 3' strand does not act as a template. It is not the template strand for synthesis of RNA. So only one strand of DNA double helix is used for transcription. Transcription means synthesis of messenger RNA. Now in the process of transcription, 3 genes are involved: promoter, terminator, and the structural gene. The structural gene is the segment of template which is transcribed into messenger RNA. The structural gene is the segment, it is the region, it is the sequence of DNA which is transcribed into messenger RNA. So structural gene is responsible for the formation of messenger RNA, and that messenger RNA will be translated into a polypeptide chain. So this structural gene is expressed in the form of a polypeptide chain. The polypeptide is responsible for our traits, our morphology, our phenotypes. That means the structural gene is responsible for the expression of our phenotypes, morphology, or our characteristics. So this is the structural gene which lies between promoter gene and terminator gene. Structural gene is the sequence of DNA which is transcribed into messenger RNA and translated into a specific polypeptide. So structural gene is expressed. Now promoter gene, the structural gene, and the terminator gene, they together constitute transcription unit. Promoter, terminator, and the template strand, the structural gene. Only the structural gene of the template strand, only this segment is called structural gene. The promoter, the terminator, and the structural gene, these 3 specific sequences of DNA together constitute a transcription unit because they are involved in the process of transcription. The enzyme RNA polymerase, which catalyzes synthesis of messenger RNA, binds to promoter. So promoter gene signals beginning of transcription, initiation of transcription. It is the gene to which RNA polymerase binds. It provides binding site for the enzyme RNA polymerase. Terminator is the gene that signals termination or end of the process. When RNA polymerase reaches terminator gene, the process of transcription is over. The process of transcription, the process of RNA synthesis ends. So terminator is the gene that signals end of the process, and the promoter is the gene that signals initiation of the process. The promoter is the gene to which the enzyme RNA polymerase binds so that it can initiate and start the process of transcription. And structural gene is the segment which is transcribed into messenger RNA. So this is the transcription unit.